This is Catalina Bustos, and today's presentation will be exploring the transformative technology of DNA editing, its application across ecosystems and generation, the opportunities, the risks, and the ethical responsibilities it represents. We'll be going over about what's DNA editing, the application is ecosystem preservation and food security, the impact it has on human health and reproduction, the safety and ethical consideration, as well as global regulations. Introduction to DNA modification technology. We need to start going over what's DNA editing and how does it work. DNA editing refers to the targeted alteration of genetic sequences between an organism's genome. Techniques like CRISPR allow scientists to add, delete, or modify DNA segments, enabling control over traits and functions at a molecular level. Major DNA editing techniques. CRISPR Cas9 is the most prominent DNA editing tool known for its accuracy, efficiency, and ease to use. Other technologies include TALENT and zinc finger nucleases. Cas9 is the easiest because it can be programmed to follow a guide RNA, and it can cut pieces of DNA and bind together the target DNA with the guide RNA. It's very interesting. On the other hand, TALEN is a molecular binding domain engineered to recognize and bind to any DNA sequence. It uses a full nucleus that codes both strands of DNA. It gets introduced into a cell where it identifies and cuts DNA, and then the cell's natural repair mechanism fixes the break, and the zinc finger nucleus is a zinc finger protein that's made of modules that can recognize between 3 to 4 base pairs that target long sequences of DNA. Its main use is in human gene therapy for sickle cell anemia and HIV, though it can also be used in plants and animals. It also uses a phosphate nucleus, which is fused with the zinc finger protein and cuts when the two zinc finger nucleus bind together. Um, then we have the application in ecosystem and food security, restoring genetic resilience in endangered species. By editing genes, scientists can increase the resistance to diseases and environmental stressors by modifying crops and organisms to enhance them against climate stressors. These increases can be from heat tolerance, pesticides and diseases, drought, nutrients, carbon captures, especially to replace genes or add beneficial traits or speed up processes. addressing food security challenges. Through enhanced crop nutrition, reduced spoilage, and improved adaptability, DNA editing can contribute significantly to global security of food and nutrient improvement, especially by building genetic diversity, making adaptive traits, or as well as stronger organisms, or restoring lost genetic diversity, controlling invasive species, and improving monitoring of endangered species. Impacts on human health and reproduction. DNA editing offers medical breakthrough by preventing genetic diseases and advancing reproductive technologies, but it also raises complex ethical questions about human enhancement. Preventing severe diseases. Gene editing can correct mutations responsible for inherited disorders, offering hope for cures and healthier future generations. Um, it is also very beneficial. It can help eliminate disorders such as cystic fibrosis and sickle cell from future generations. It could also be beneficial against infertility. Um, DNA editing techniques may assist in overcoming infertility issues and improving reproductive outcomes, though clinical use requires cautious evaluation. Um, it can help against infertility. It can also make uh, higher pregnancy outcomes, especially in agriculture. In vitro can help with DNA editing, can make healthier babies, and it could also reduce the amount of money that farmers will spend on their animals. Um, as I said before, it could help for infertility issues, as well as any preventive disease. Uh, but this can also be concerning because it could lead to uh, designer babies, and it could also have a probability of off-target consequences, and in some places, human uh, DNA editing is still illegal, and these procedures would more likely use the CRISPR-Cas9 method. DNA editing is safe and ethically applied is critical to prevent unintended harm and respect societal values and future generations' rights. DNA editing may introduce unintended genetic changes, and the long-term effect on ecosystems and humans still remains uncertain. It is important It is very important to continue to have researches and investigations regarding the probabilities and possibilities that DNA editing has in medicine, agriculture, and technology itself, though it's very important to have a record of any possible mutation and off-target procedure because it is still new. We don't know how these mutations could affect future generations in case it is applied. So it is very important to keep this in mind while making legislations to regulate these techniques. Modifying germline DNA affects not just individuals but their descendants, raising questions about consent, naturalness, and responsibility towards future generations. Because we still because this is still a new technology, we don't know how it could affect at a long term, and so it is very important to legislate having that in mind because we don't know the extent of possibilities that DNA editing has in uh, our lives now, but these can also evolve, and so it is important that more than one area of government is having consideration in these topics. Global regulation and responsible use. Government and international bodies must establish clear policies and frameworks to guide ethical, safe, and equitable use of DNA editing worldwide. Uh, if there is no regulation, uh, scientists could experiment with anything and anyone, and so it is important to have a guideline towards what is okay and what is not okay in case of any wrongdoing. Importance of regulations. Effective regulation ensures that DNA editing research and applications adhere to safety standards and ethical principles, protecting public health and the environment. As of today, there is no legislation on the US limiting the type of work of DNA editing, though germline DNA editing is illegal in the US. There is only approved use with research and medicine and agricultural procedures. Um, but it is important that there is an institution that only focuses on DNA editing and the practices are going on worldwide. Transparent decision making and efforts to provide fair access can build public trust and prevent disparities in who benefits from DNA innovation, especially because this is a new technology. Any kind of marketable procedure would be very expensive, which would limit the amount of people and the kind of people who will be able to access from this technology. Lower and middle class families would not be able to access such technologies because they will not have the resources to do so. So in case this gets able to become legalized at any way, it should be the government should be able to provide a way to all kinds of families or income people to get access to such technology. DNA editing presents unprecedented opportunities to transform ecosystem, agriculture, and human health, but demands careful consideration of risk, ethic, and regulations to realize benefits responsibly. There's two main takeaways from this, and there is still room for improvement in this technology and to have new ways of uh, doing DNA editing, but there is a need for regulations to do so because it is still new there is some regulations, but there needs to be a worldwide institution that only focuses on these topics because it is very new, and so it needs all kinds of people with different knowledge and education to go through these often, not just once in a while. And so, yeah, this was my presentation on DNA editing. This is Catalina Bustos, CCM class of 2026. Thank you so much. Have a good day.